Okay, thank you, Dr. Fink. Thank you, in-person attendees. It's 2025, and thank you, virtual attendees as well. We hear you, we see you, but not until the end. Okay, so I am super excited to talk to y'all today about plasma and CSF metagenomics. We're going to talk about metagenomics broadly speaking and drill down. This is a problem that is near and dear to my heart. How do we use these tests? It's incredibly disruptive and a stewardship success story. I was going to say nightmare, but I changed my mind. Here's my disclosure statement. I've worked with a couple biotech companies, nothing related to metagenomics at all. Done a little bit of an insurance consultation. And here are my 3 learning objectives. They're a little bit wordy up here because when you're looking at a screen, you want to be in depth, but I'll tell you what they mean in simpler terminology. First, I want to have you be able to tell me what is available for clinical metagenomic options, what you can actually order today, and what are the different types of characteristics that these tests have. A lot of the basic details get kind of lost when I say, can't we do metagenomics on that? And you're like, sir, that is a toe. Next, we're going to talk about some of my favorite and then less favorite indications for when you should try to employ plasma metagenomics to your clinical use. And we'll look at some of the data described in the literature and maybe some places for improvement or places we wish we knew more. And then lastly, we're going to talk about how we developed a policy at Children's to kind of get everybody on the same page about how these tests should be used and to make sure that the folks who are best at interpreting infectious disease diagnostics, hint, infectious disease physicians, are kind of driving the process and interpreting things. Okay, so let's just jump into it. It's been a big year, 2024. There are all kinds of news headlines coming out. Carius, who's your big major corporation for plasma metagenomics, is getting FDA tags for breakthrough testing. They're expanding through BARDA contracts to look at RNA organisms as well as the DNA organisms they already do. For CSF metagenomics, you're seeing that Mayo Clinic is now offering their own assay because UCSF has kind of handed off their mantle as the one offer of CSF metagenomics. And then there's another startup company that kind of spun off of UCSF that is trying to take on CSF metagenomics and turn things really, really fast, where typically is a very slow test. Not only are we seeing these changes happening in the ecosystem, but also looking at the amount of cases we're seeing at Seattle Children's where people are asking for this test. We were really starting to hit that exponential phase. I started in, I think, July 2021, and I was like, oh man, like we're getting so many of these. And now, I'm having to deal with like 3 a month, which I imagine the number is going to go way up over time because we're still figuring out how to use things. So it's been a big year. So let's start with the basic question. What is metagenomics for the diagnosis of infectious disease? When you see MNGS, I'm saying metagenomics, metagenomic next generation sequencing. So I'll just call it MNGS. Really, we're only thinking about infectious organisms here, but of course, everything in your body is creating cell-free DNA in one way or another. So all living things that we care about, when they die, they release some sort of nucleic acid. Here's a little diagram just showing a bunch of things in your own blood that might release cell-free DNA. Of course, we don't really care about this stuff, so we'll talk about how we'll filter it out later, but the microorganisms inside us are also releasing their nucleic acids when they die. It's worth noting that compared to how we think of as maybe full DNA, DNA coming from cells, the DNA that we see cell-free is fragmented, highly fragmented. And so this is just a chart looking at cell-free DNA in human urine and just showing those size of the fragments in terms of base pairs. So the biggest peak is 250 base pairs, which is terrible. Even if you knew your target and you knew it was in there, if you're just fragmenting every 250, you might be cutting right in the middle of your PCR primer target. So really what we're looking at is much different than when we extract from whole cells. At a very high level, the data analysis pipeline, this is taken from Carius's website, describing how they do it. So plasma is their specimen of choice. They process their specimens, including nucleic acid extraction. They kind of brush over the fact that there's a huge host depletion step, or at least, they may not publish it, but it has to happen. We're really looking for that needle in a haystack. If we look at in your body, you're going to be mostly human cell-free DNA, but we don't care about that. We want to find something else that's there. Of course, you do sequencing, and then you can further, pull out all the sequences that match to humans, or maybe some plant material that we don't care about unless we discover something new here. And then, of course, we have a data analysis pipeline. We're putting it all together, saying what we actually saw in that sequencing, and then we report it to you, and you can find out what is actually in your specimen. So when you see these terminology, we talked about metagenomic next-generation sequencing. You might also see me say microbial cell-free DNA or MCF DNA or cfDNA, just talking about the actual analyte that you're measuring versus metagenomics is the technique that we're using to analyze it. So at a very high level, why is this interesting? Why is this method even being considered clinically? So when you think about conventional methods, you're talking culture or PCR, maybe serology. These are relatively hypothesis-driven in the sense that if you're thinking you're worried about acid-fast bacteria, you order an acid-fast bacteria culture. The cost is relatively low. Some of the methods are quite old. Culture media is quite literally grows in the ocean. We're just using agarose. And then when you grow it in culture, you can perform additional testing like susceptibility. You can look for phenotypes that you can't necessarily do even with PCR, but also with sequencing data, although we'll talk about how that's changing a little bit. Pretty established significance too with the testing that we know, and testing is local. It happens usually down the hall from wherever you're collecting the specimen, down in the lab nearby. A metagenomic method, on the other hand, is going to be, they like to throw this term around, agnostic. And I think it's worth kind of drilling down on this term for a second, which agnostic doesn't mean that you have no hypothesis about what results that you might have. Agnostic really just means that it's going to look for anything. And if you didn't include a specific PCR primer set, you're going to miss it in a conventional test. But for metagenomics, you would still find something. Now, what you do with that information, it may depend on what it is. You might get a virus you've never heard of before, and I'll maybe throw one or two of you out there today. In terms of cost, it's really expensive. For plasma metagenomics, your commercial cost is $2,000 per specimen, and your cost for CSF metagenomics is closer to $3,000 per specimen. These are never reimbursed by insurance payers. They're considered experimental at this time. Hopefully, we start to see some medical necessity policies, but they're just not there yet. You can get a little bit of susceptibility information if you get lucky and you catch maybe carbapenemase or something like that. But it's really hard to say if you don't catch it, does that mean that you're susceptible? And maybe we just need more information on genomics, but that'll come with time. Interpretation can be different. We'll look at a bunch of cases where you're going to get a bunch of stuff and wonder what the heck you should do. And of course, this is not really happening locally at very many institutions. Some institutions are developing their own metagenomics assays, but it's taking some time, particularly with FDA regulation coming down the line for LDTs, laboratory developed tests. It's unclear what the leeway of your local microbiology lab will be to create a test like this. And so the big question, of course, is when should we use metagenomics versus conventional methods, or how can we use both in perfect harmony? And so we'll get there. In terms of challenges of this testing, generally speaking, one thing that makes this test hard to interpret is that analytically, it's incredibly sound. If you sequence and you find strep mitis DNA, you know that strep mitis DNA is there. Say it's in your plasma. But what is the significance of DNA in plasma? It's kind of unclear. Are you catching it because maybe somebody has a breakdown of their oral mucosa and you're just detecting like a mouth commensal organism? Or is this causing a deeply seated infection in your brain and the DNA is leaking out? It's hard to exactly say. In terms of sensitivity is imperfect. It doesn't detect every single infection. We'll look at some examples of how it stacks up against conventional testing. There's not a lot of clinical outcome data. So going back to what does DNA-emia really mean? Well, the data on, what if you treat everything as real and you treat everything you find in metagenomics, how do those patients fare versus when you just look at conventional testing? The data is not really there yet. There are some folks who are working on it, and it's in process, but best we have is sort of imperfect methods of adjudicating how much value is added to patient care, which kind of leads us to this last point, which is, when we get these results, what are we doing? Maybe we're acting differently. We're using a different treatment. Maybe we're deciding to discontinue pursuing infectious etiologies at all. We're changing our behavior in some way. The question is, are we changing our behavior for the right reasons? It's hard to know without that clinical outcome data. Now, you could say, well, we have a new result. Not all new results are specific. You might be picking up a commensal organism, or you may be confirming a diagnostic result, which holds really high value for some certain patients, but not necessarily for everybody. So there's some tests that we know those results and we can trust them. And then, of course, if you want to really drill down to it, the best we have is kind of like an infectious disease physician sitting down and looking at everything and being like, this makes sense. This is a likely cause of this patient's infection, even though we didn't catch it on culture or PCR or anything, or this doesn't make sense and we still think the patient has an infection. So it's really hard to figure out what is truth. There's no gold standard comparator. Okay, so that's a whole lot of high-level stuff. Let's talk about nuts and bolts of what tests are actually orderable. So we'll start on the left. For plasma metagenomics, this is offered by Carius, the commercially available option. A few institutions might be working on local testing options, but not widely available other than Carius. It can give you plasma, in a PPT or EDTA tube. You can freeze it, which is great. We advocate you freeze, collect, freeze, and then we can talk about it as time goes on, because the earlier in infectious disease course, usually the better your specimen is in terms of diagnostic yield. They have a very impressive 48 to 72 hour turnaround time. They'll tell you 48, but you want to take into account the fact that you got to FedEx it, and you might only have a FedEx once a day or something like that. And then it's also, for now, inclusive only of DNA pathogens. So this does matter if you're looking for, say, measles or something. You would miss it. In terms of CSF metagenomics, now on the right side of here, actually both our testing options are brand new within the past year. Mayo started offering their test earlier last year. Still a 1 to 2 week turnaround time. They'll take just any CSF and a sterile cup that's been frozen. They'll also take it refrigerated. So again, you can hold it for a long time before you decide if you want to test it. And then a new company, Delve Bio, which sort of spun out of UCSF's old assay and has some relationship with them, although they're located in Boston, which I found out the hard way when I tried to send them a specimen during a winter storm, and I got stuck in middle America for a couple days. And they are promising a much faster turnaround time. This is brand new. This just happened in December. And so we're still kind of trying to figure out, how does this fit into our testing algorithm now that we can have these results that historically took 2 weeks in 2 days. And we'll talk about some of the implications of that. So broadly speaking, for these two options, when are we supposed to use metagenomics? We can look at what the manufacturers of these tests say. So CSF metagenomics, this one might be a little more clear. If you suspect a central nervous system infection, you should, and you can't diagnose it through other means, maybe consider testing of the CSF. This is useful in the sense that, you're pretty sure that normal CSF doesn't have any DNA background in it, unlike blood. And so if you find something in the CNS, you can suspect that's actually causing the CNS infection. A little bit closer cause and effect. Not many indications. Meningoencephalitis. Plasma metagenomics has been broadly applied to many different types of syndromes. It was originally approved for sepsis, but seems to be less used for that nowadays, although you definitely still could. Big one is febrile neutropenia. We'll talk about the ones in bold just because they happen often at children's. Infective endocarditis is also a big one. There's actually great data that shows the duration of nucleic acid material in infectious endocarditis patients. Fever of unknown origin. There's another one, invasive and fungal infection, which we'll talk about, pneumonia, and many, many more. Of course, if you're looking at this and being like, well, it's plasma. How do you know it's coming from the heart or the lungs or, maybe a deep-seated infection in your bone or something? That's a good question to ask. And indeed, if you look at Carius's own validation data, they show in a healthy cohort here on this graph on the left that you actually still see some detection of microorganisms in patients who are without symptoms. They quantify their the amount of nucleic acids they see, and kind of comparing them on the right, there's a lot of overlap between what is considered to be a definite pathogen and an asymptomatic individual in terms of the quantity of the organism too. So these results are not straightforward to interpret. They can absolutely provide value, but it does take a keen eye, and you have to apply it in the right situation. And so this is just a way of saying cell-free DNA doesn't mean infection always, but you should really think hard about it. We see this reflected in our own data, so I have this kind of broken down. Hopefully y'all can see the colors just fine. They're not too washed out. But looking at metagenomics results at Seattle Children's, we're lumping CSF and plasma together, where we have our definite pathogens at the very bottom of our pyramid in green. We just had one per year. We've had a couple that are maybe probable or likely potential pathogens, but we couldn't say for sure in red, so 3 of those. And then we get a lot of what we've just been calling insignificant detections. These are a lot of commensal viruses, CMV, EBV, BKV, sometimes normal gut flora where, well, maybe it could cause infection, but really we were actually looking for a fungus in this particular patient, so it doesn't really help us there. And so most of the time, our results are positive for plasma metagenomics, but the actual results are not really actionable. This is kind of flipped. I didn't put it in here for CSF, where our results are very rarely positive. We'll talk about it later, but when they are, you know exactly what's going on. I'll throw out two exemplary plasma metagenomics cases because we're going to talk mostly about plasma metagenomics today. And so really, we like to think of these cases in three sections. How we choose the case, how we plan to interpret results, and what actual impact we can bring out of testing. So this is what I consider to be a good case to start with. So we had a patient who has this congenital heart malformation. He has a prosthetic valve. He's had these inflammatory markers and some evidence of myocardial injury. And our infectious disease team is trying to figure out whether or not this kid has infective endocarditis. It's a long course of antibiotics if you do. But our other markers and way of adjudicating that, not so good. We felt like we couldn't say one way or the other. And so we came up with a pretty, I think, solid interpretive plan. If we found a bacteria that is associated with endocarditis, boom, we'd say that's good reason to treat. If we found a commensal organism, then we'd say, well, that's not really an endocarditis bug. Maybe it would kind of weigh our treatment decisions a little bit. Maybe you'd want to include it in coverage, even if it did feel weird. But if you had a negative result, they were saying, well, there's nothing there. We're not worried about infective endocarditis. And that's what happened in this patient's case. We were actually able to discontinue, and the patient did just fine. So a way of kind of getting out of that conundrum. Conversely, we had a little bit more of a tricky patient here. We had a bone marrow transplant patient. He's been having ongoing neutropenic fevers by antimicrobials, and we're trying to figure out, is this kid experiencing an infection? Are we covering with our empiric antibiotics what he's being infected with? And we end up getting a result of primate Boca parvovirus 2. I didn't know about this virus until I read it. It turns out you can blame the taxonomists. We are indeed primates, and so this is not acquired from non-human primates. It's just found in humans. And it's ambiguous clinical significance. Maybe it has some GI elements, but it doesn't really answer their question with this patient or what to do with this result. Well, we didn't do anything different. And so I'll give that a little face palm. Okay, so at a high level, we talked about the two types of tests that you can order and the slight differences between them. But now let's talk about where there are advantages or potentially disadvantages for conventional testing. So we'll start with CSF because I think this is a little bit more straightforward. Indications are limited. You have meningitis, you have encephalitis, maybe you see a brain lesion. You're expecting infection in the CNS. You're not going to see normal flora unless you mess up your collection and accidentally get a little staph epi during your LP. We have good markers for what CNS infection looks like. We can look at CSF parameters, your glucose, your protein. We can look at cell counts. So we can have an idea of what we're looking for ahead of time. And also to tell us, we've done PCR, it's negative. Maybe we should consider looking for other things that wouldn't be captured by PCR. But our testing yield has been low at Seattle Children's, and I'll show you some data from UCSF. It's kind of been low generally, but there's opportunity for improvement. In terms of who tests, we talked about Mayo, we talked about Delve. I think that there's not a lot of data comparing the two because they're relatively new, but they're probably analytically both very sound. The time is, of course, huge, and at a similar cost point, really that it's hard to turn down a 48 to 72 hour turnaround time. And then I have UCSF up there. They've hung up their hat. They're sick of children's hospitals from around the states calling them, being like, "Can I expedite my specimen, please?" But as they hung up their hat, they did release a wonderful article saying, what did they see as the single offer of this test for 7 years? What did they actually see? And so I'm going to throw up some charts. They look information dense, but we'll just talk through them. And so this is actually taking into account the organisms that they actually found in total. So including things that were already known, although they're reference labs, so they don't necessarily know what you already know. But broadly speaking, you're seeing things that you would expect to see in the CSF, DNA viruses like HHV6, EBV, you're seeing West Nile virus, HIV, which it seems like most of the time, as we'll see, that folks already knew that they had HIV. You're also seeing some more esoteric stuff down here in your fungi, which is definitely some of these fungi are never considered to be normal flora. You see aspergillus, you see histoplasma, that should not be there ever, even if it's just DNA only. Some parasites were found as well up there, and then a whole lot of other viruses, enteroviruses, echoviruses, things that maybe we don't have targeted testing for. It might be missed by other assays. It's also worth looking at what they found by metagenomics alone, and that's what I have on this slide here. So here you can see a lot of those kind of like large groups have gone away, and that's because conventional testing is able to pick up the bugs that we know, the organisms that are kind of on our standard treatment workup. We'll talk about a little bit of the tiers of encephalitis diagnostics. And so you see much fewer from that standard workup list. Now you see a little bit more serology-focused organisms. So maybe you're looking for that organism in a different way. You were looking for serology before, but now you're actually looking for the presence of nucleic acid. So maybe detecting something before a serologic response. You also see a smattering of bacteria here. All of these are probably significant. And maybe those are below a limit of detection of other assays that we have, or maybe you actually didn't pursue broad spectrum PCR. It's kind of hard to know in terms of what these reference lab samples did. But interesting to see. It picks up a little bit of everything. It could be useful. It's worth highlighting that they also looked at the specimens that came from within their own lab, and about a little over a third of them were only diagnosed by metagenomics only. So just kind of summarize what we know about the performance. Overall detection rate at UCSF, everybody's specimens together, was about 14%. They looked at a smaller cohort, but can probably be extrapolated to a larger cohort, where if you take into account conventional testing, your diagnostic yield is probably like a third or so of that. So maybe like 5% of the time you're finding a new organism. In practice, it's really hard if you have a patient who has a CNS infection type picture to say, okay, we'll tell you in 2 weeks. By then, you may have already kind of resolved the clinical infection, or you may have decided that it's not infectious. And a lot of cases at Children's, that's what we saw, which is not really any follow-up when we got the result. We didn't have a lot of positives, but negative results weren't that impactful. So really, it's this exhaustive testing prior to doing metagenomics that might be taking away the diagnostic value that it's actually added. So I'll share with you just a quick typical metagenomic CSF case, just so you get a flavor of what we're looking for. So we had a young child, not immunized. They had 3 days of altered mental status. They were vomiting. They had some fever. Some imaging and labs, preliminary labs, made them think meningoencephalitis. We didn't have a good infectious workup, and we also didn't have a good immunologic workup. And the team was saying, hey, we need to manage these symptoms with steroids, prevent the inflammation. But if there's a raging infection, we don't want to slow the body down in fighting it. And in this particular case, although it took us a while to get there, we got a negative result, and that was able to let the team move on with steroid therapy. So this is a case where we actually got some, but unfortunately, a lot of the time we don't actually get a clinical action that we want. Okay, so let's talk about tiered workup. Really, with encephalitis workup, you want to get things fast because this is one of the most sensitive places to have an infection. So your first tier, things that you can do at your own hospital that you can do quickly. So doing a gram stain of CSF, doing a cell count. Maybe you can do bacterial or fungal culture that might, we might have an answer in a day or two if they're pre-treated or not pre-treated with antibiotics. You also are probably going to do a rapid PCR panel. We know what the common organisms are, and common things being common, we detect them quite often, but those uncommon cases are still really impactful. And so our second tier is a little bit more kind of choose your own adventure type of testing. You might do broad spectrum PCRs. You might look for some non-specific fungal markers. You might look at, investigate TB serology. You might look for specific viruses, PCRs. You might look for arboviruses. There are a lot of things you could choose. And we often don't do everything, but we just kind of pick what's likely based on where that patient has lived. And so we're going to talk a little bit about cost and how maybe, this test is analytically sound, doesn't have high yield, but maybe can replace some of these more esoteric tests in terms of cost. And so there's a slide put together by Caleb Stokes, one of our ID physicians at Seattle Children's. And he figured out the costs of all of our tier 1 and our tier 2 meningoencephalitis testing. And then up here in the top right, we have what's the cost of UCSF metagenomics, which is roughly the same as Delve or Mayo. And what we found is that, our initial cost is quite high. It's worth noting that our rapid PCR, that's like the build cost as opposed to what it actually costs the lab. Whereas when you're sending to a reference lab, the lab has to pay that cost. That's not the cost of reagents. So it's a little bit, in total, a little bit higher to send out. These things it's hard to ever get away from at your hospital because you need that information stat that can help a lot of patients. But really it's this tier 2 where everything else in your tier 1 comes up negative. And you go, well, what can we look for? And if you don't have high suspicion for JCV or amoeba or, maybe toxoplasma, is there value in maybe kind of lumping them all together and just say, hey, well, we're going to do a metagenomics assay and then say, we look for all of them. Maybe you pick your favorite 2 that you think they have the highest risk for, just because single plex testing tends to be more sensitive than broad testing. And if you stack it up, you could say, hey, well, if you followed our algorithm to the T, you would get a decent cost savings looking at tier 2. Now, the hard part up until recently has been, well, you have to wait 2 weeks. And you're definitely not going to sit on your hands and say, well, we're not going to send any testing because we're going to get results in 2 weeks. And so at our institution, we haven't been in a place where we could ever do that. Now, with the 48-hour turnaround time of Delve, we haven't sent them many cases, maybe just 1 at this point. But we're hoping to say, hey, can we trust the results? Can we feel good about those results enough to maybe not look for everything piecemeal and order them in bulk, Costco style? Okay, so in general, we think that maybe metagenomics of CSF can add value by knocking out large swaths of specialized testing as opposed to, kind of being end-of-line testing where you've already ruled everything out. If you have that faster turnaround time, maybe we can get more value sooner. But there's currently not a lot of consensus on when should you send. What is that trigger in terms of time or patient presentation or lab values that cause you to say, now is time for metagenomics? I'll show you what Children's has kind of implemented at the end, and then maybe we'll see if that kind of works. Okay, we're going to run through these relatively quickly because there's a lot of stuff for plasma metagenomics. Mostly, I just wanted to show you a million different things about all the indications to confuse you because it's confusing to us too. And we're trying to pull that apart through stewardship. But in terms of your testing option, really easy. You got one lab, it comes quickly, if you can get it to them quickly, and you can freeze a specimen, hold on to it for a very long time. And most importantly, it only looks for DNA, at least for now. They're working on expanding it. So let's cut to the chase. There's one meta-analysis out there by Denver Niles, and he looked at all of the single-site studies at that time. It was published in 2023, and tried to figure out what the positive percent agreement was and the negative percent agreement was between plasma metagenomics and a composite review of not only your actual testing results for standard microbiology testing, but also did an ID physician think they have an infection or not. And so basically on this chart, you're looking at PPA, MPA, PPA being plasma metagenomics had a positive result and you think your patient has an infection with this organism. MPA being this plasma metagenomics was negative and you think your patient doesn't have an infection. So basically the more far you are on the right you are, the better. And what you can see is that between the different sites here is vastly different. You have some that are approaching near 90 for PPA, and you have some that are down maybe around like 40-something%. And there are a lot of differences between these institutions and how they handled who was selected for plasma metagenomics and how those results were interpreted. And indeed, actually, if you end up looking at these single-site studies, they feel a little bit differently about how their results worked. Actually, Hogan, I think, felt like their results were worse than what is reflected in this graph. And some institutions felt like their data was much better than this. But it all depends on kind of the eye of the beholder. What are you doing with that information? How do you think it fits with that clinical picture? So there's a lot of wiggle room for interpretation. The key factors that are worth talking about, because we're going to come back to them when we talk about stewardship, is that if you have lab and ID involved, if you use conditions that we know a lot about, and if you learn from your own mistakes, you're more likely to push to that right side. So a lot of the early publishers of their findings are kind of more on the left side, not so good testing. And then as we learn more, we're being pushed to the right. So try to figure out what lessons we can learn. The other thing you can think of is that, here's a slide from Denver Niles' single site study, is that maybe it's really just buried in the details. If you lump them all together, it's hard to say, is it useful, is it not? But you should really break it down by why you're doing the test. You can think about it as many different tests depending on what your patient is actually experiencing. So, deep infection, neutropenic fever, sepsis, culture negative endocarditis, those sorts of things. And so we'll talk about some of them. Febrile neutropenia is one we talk about often, just because we have a lot of patients who are neutropenic and have fevers. This is data, I believe this is from, or at least co-authored with Carius, but just looking at when you do plasma metagenomics from patients who have neutropenic fever, how often do you come up with something new? And the answer is almost twice as often you come up with a new result. This is a little tricky. Here's a kind of an uninteresting list of all the organisms that were seen in a cohort of pediatric febrile neutropenia. But you see a lot of organisms that you might expect to see if your mucous membranes are breaking down or your gut isn't completely intact. Maybe you have tonsillitis, something like that. You're not feeling so great. A lot of E. coli, a lot of anaerobes. You see a few in there that are definite pathogens, like a rhizopus, like you never want to see that type of fungi in your patient. But, a granulicatella, I don't know. It could be coming from your mouth. But it could also be causing infection. We don't really know. I think this stresses the importance of strong empiric therapies, that you have to cover what you might expect to cause neutropenic fever, what you might expect to cause infection. And you can't be confident that any one test is going to tell you yes or no. So going back to that healthy cohort, again, you see that the organisms that you might find, again, like gut commensals, you see oral commensals, strep mitis, Neisseria species. You see normal human DNA viruses. There are just many things that are common to be seen. Running through a couple quick febrile neutropenia cases. First, we have a patient with BLL. They had this neutropenic fever. They have DRESS. They have an abdominal distension. They're being empirically treated with metronidazole because they're worried about gut stuff. And they felt like if they had a plasma metagenomics result, they could make sure that they're treating everything. This was one where we kind of got everything in the kitchen sink. We got club pneumonia, E. cloacae, parainfluenzae, some HHV6. And we weren't really able to escalate antimicrobials. We were already covering everything. And we weren't able to de-escalate antimicrobials either because we were like, well, what are we missing? What else could be there? Is there fungus? Is there something else? So that was a tough one. It's hard in these critical cases where you feel like you have to look. Here's another one. It's a little bit of an oddball for us. We had a pre-BMT patient, neutropenic fever, cervical lymphadenopathy. They had some pulmonary consolidation. So, maybe respiratory. They're just kind of looking generally for infection. But interestingly, they had already had 2 plasma metagenomics tests that showed Stenotrophomonas multifida during a neutropenic fever. So the team was like, we want to look and see if she has Stenotrophomonas multifida infection. Sure enough, we found it. But it's really hard to say whether we could have stopped treating using Bactrim for Stenotrophomonas multifida with a negative result. It's also hard to say whether there is an infection. Nucleic acids can persist for a really long time after infection is resolved. So not clear benefit to the case, but we felt compelled to look. I think it's worth taking a look at the test report just because Carius has listened to folks who are worried about commensal organisms. They actually break it up now into 2 different parts, showing, hey, this is an obligate pathogen, professional pathogen, this is Mycobacterium tuberculosis. But then they say, well, this is commensal pathogen, so maybe take it with a grain of salt. So they're working on helping interpretation. Okay, so for bloodstream infections, it was originally validated in sepsis, but actually they felt like they could predict bacteremia possibly even ahead of time. Here's one study that showed that if you've grown an organism in blood culture, you'll actually be detectable via plasma metagenomics far after you've been blood culture negative. So it's good evidence. If you're looking for an organism in the blood, I think it makes sense to do plasma metagenomics to see if that organism is in the blood. Maybe you missed that period earlier on and the patient's now pre-treated and you don't expect anything to ever grow out of blood culture. So there's some value here. I think it especially lends itself to infective endocarditis, which in this particular cohort, again, we're looking at red bars being blood culture and blue bars being cell-free DNA, is that cell-free DNA persists long after the organism stops growing in culture. So if you're suspecting infective endocarditis, you might catch it via plasma metagenomics. There have been a few larger cohort studies. There are kind of 2 branches here of these patients who were ultimately determined not to have infective endocarditis and those that were. What's notable is that some of the no endocarditis patients still had a positive from plasma metagenomics, and some of the definite infective endocarditis patients still had a negative result. So, now actually plasma metagenomics is mentioned as part of the Duke's criteria. I forget which level exactly is that, but this is one of those where you kind of have to take everything in the constellation of symptoms and evidence that you have for a particular patient. So maybe a piece of evidence might not be the only definitive evidence you had, which lab folks like myself don't like that. We like a positive test equals infection and a negative test equals no infection. Infectious disease physicians tend to be a little bit better at Bayesian reasoning, be like, okay, that's 60% more likely. Lastly, we'll talk about invasive fungal disease. I think this is a great indication, particularly with some of the patients that we have. I know some of our colleagues at the Hutch use this for this reason. So most fungi, they're not going to be normal flora. If you see a mold that really shouldn't be there, nobody's colonized with molds, at least not that we think. So your specificity is just going to be a bit higher if you restrain your analysis to just looking for fungi. If you have a positive result, usually it's going to have an action that you're going to take, or it's going to really confirm like, hey, we really need to stay on these antifungal drugs. Conventional testing is not great. So we have some non-invasive tests, some biomarkers of fungal infection, but they don't catch everything, and we're pretty familiar with the fact that they're insensitive and imperfect. And you might not be able to get an invasive testing option. You might not be able to sample a deep tissue site because some of these patients are pretty unstable. For a reason, we also think that the specimen quality is maybe less affected by pretreatment. I'll show you a slide on that. And we have pretty good established criteria for what is a probable and possible invasive fungal infection, because we're really used to patients who have probable invasive fungal infection never actually grow an organism. We're just kind of working with like just the ghost in the background. And we'll come back to some of our primary review criteria, but this fits really well with our stewardship goals. So our colleagues at the Hutch, this is from Joshua Hill, they put together and looked at a retrospective cohort, and they found that actually if you pair looking at galactomannan with plasma metagenomics, you actually catch a really broad amount of individuals who have probable or possible, probable, or confirmed pulmonary invasive mold infections. They broke it up a little bit further, looking at the different types of organisms that you might think of. Aspergillus, of course, we have specific markers, Aspergillus galactomannan, where for non-Aspergillus like Mucor, we have fewer markers. Maybe you do beta-D-glucan, but not everybody creates beta-D-glucan. They actually found that the yield was a bit higher in these non-Aspergillus organisms, maybe because the markers aren't as perfect. What is notable is that if you look at possible invasive mold infection, this is basically saying, hey, we looked at everything, we did all the testing we could, and we just couldn't find an organism, that the yield was pretty low, at least in their cohort. Now, this might be because maybe they actually, maybe it's not possible after all, maybe they don't have infection, but it is also likely that if you have an organism that is has a focus of infection that is well circumscribed, it's kind of walled off, maybe you're just not getting that cell-free DNA out of there. Interestingly, if you there's a group out of the Netherlands and Belgium, and they're actually looking at comparing similar type of case, looking for invasive mold infections, looking for fungi, but comparing plasma versus BAL, so direct sampling. Maybe you're not taking tissue, but a little bit more invasive, but kind of makes sense. Now you're looking right where you suspect the infection to be. And if you look at kind of these little yellow wheels here, you see that the area marked in blue is what was found by metagenomics, and it increases quite a bit. This makes sense. If you're looking for infection and you look at the site where you think the infection is, you're probably more likely to catch the organism. And then last, this is, maybe not necessarily proven data, but kind of interesting, is that over time, there's a group who found a low association with the fact that maybe actually the amount of cell-free DNA goes up during a fungal infection. And this might make sense as you get more dissemination, more tissue destruction. So maybe a good target for those reasons. Okay, we'll blaze through lower respiratory really quick. This is just kind of looking at BALs or versus BALs. Everything in blue here was detected by BAL. Everything in orange was detected by, BAL conventional testing. Everything in orange was detected by plasma metagenomics. There's significant overlap. It's worth noting that it misses a lot. So a lot of things that you would only have by conventional testing are found, but there are also a fair amount of things that are only discovered by metagenomics. And the question is, are those significant? Is that causing our infection, and should we treat it? And sure enough, if you look at those organisms, some of them are clear human pathogens, PJP. If you see rhizomucor, you should be concerned. But a lot of them are potential organisms you might see in the gut, some streps, some pseudomonas, some prevotella, that sort of thing. And of course, they showed that if you paired this together, you actually get a higher yield. So maybe at least you're having a differential that's more inclusive of everything. I think we're not perfectly there for replacing BALs with plasma metagenomics, but it's something that's actively being worked on. So I thought I'd throw it out there. And there's a lot that we won't cover today. There are just so many case studies, individual things where you found something. Maybe you did it on a patient that nobody's recommending you do, but then you serendipitously found a pathogen. There's Bowel-Moving Mandrillaris that's been found, be causing brain infection. There's Leptospira, which is classically hard to identify because it's not really one of our culturables. Rickettsia, another difficult to culture organism. Coxiella, things primarily done by serology, and the list goes on and on. Atypical mycobacterium, and so there are lots of different ways you could apply this. Each application may be more or less useful based on the data we know, but there's not a lot of data that we do know, especially if we have a very specific indication in mind. And then, of course, we don't have that clinical outcome data, which we want to know. And so at the end of the day, you really have to look very specifically at your case or your patient population to come up with a plan on how do we make this result actionable? How can we maximize it? And that's where stewardship comes in. So in the last 10 minutes or so, I'm going to walk you through what we did to make this a little bit better testing at Seattle Children's. So generally, lab testing is getting more and more complex every year. There are a lot more tests. We're getting less training on how to interpret those tests, and the public is also asking for things that they may not have full understandings of. Metagenomics is a prime example of this in having difficult result interpretation. You might not know how it relates to testing we do have. It's a brand new test, and there's a fair amount of marketing pressure. And there's some shots fired in the literature too, talking about how actually this test isn't so great after all, and we just need more data. We need more clinical data. So we connected with our stakeholders at Seattle Children's. We talked to our bone marrow transplant team. They were turning this test towards the end of lives for some of their patients, hoping that they would find something that they could treat, not necessarily thinking it had optimum diagnostic value added, but feeling like they had no other option. A lot of our critical care units were also turning it to these high urgency situations where you feel like you need an answer fast. You just can't wait. But they also felt like our lab process was unclear, which is fair. I think in the early days, we didn't know what was going on either. Talking with infectious disease, who was kind of stuck reading all these results and say, what am I going to do with it? They felt that some folks were going behind their backs and deciding whether or not something was infectious without talking to them. And so it's a good reason to have ID in the room when these decisions are made, because ultimately they're going to read the report and help you interpret them. And then we also felt like a lot of these conversations were happening in person at places the lab wasn't. So we really tried to identify ID as kind of a champion of helping us how to optimize this testing. So going back to those key factors that were found in the meta-analysis, we wanted to establish some clear expectations for the ordering process, provide guidance on what was good testing, not only tell you when you shouldn't use it, but tell you this is a good time to use the testing. And make sure that experts are involved in selecting cases, interpreting results. And then also we want to monitor what's working, what's not working, what can we do better. And so we'll look at some of the data that we got today. As a big part of that, just looking at those same points are at a high level. I'll show you the flow chart, but it's kind of complex. Our flow chart here basically is we recommend good indications. If there's a potential case, we say collect first. As soon as you're thinking about it, collect a specimen, put it in the freezer, and we can send it at a moment's notice if we need to, but at least you have it early in infection. Talk to ID. They got to endorse these. And then have ID stewardship and micro meet to talk about whether these cases are useful and that we should send them or not. And then debrief after to figure out whether they actually helped you. We won't dwell too much on these pre-sending questions, but really we're trying to figure out all those things we talked about earlier. How do you change management? Are you going to harm your patient by having a result that delays a transplant? What else could you be testing instead? Are there any better options for you to test? For plasma, often the answer is no. CSF, usually there are a lot of specific tests you could add, but maybe you want to knock them all out at once. And taking a look at that large flow chart here with our recommended indications on top, telling you how exactly to save the specimen, how to get in touch with ID and talk about that request. And then for the lab, the hard part for us was we wanted to react fast. Because there's so much time pressure involved with this testing, we're saying, hey, we're going to send it out within the next window if we can, if we think that this will be helpful. And so we were meeting within 24 hours to have these discussions and really knock things out. And this was challenging to do. Luckily, we can't send on the weekends, but I guess if you have FedEx every day, you're going to have a hard time as a lab steward. This is for plasma. I should have mentioned that at the top. And of course, at the top, we say if you're looking for a CNS infection, you should probably go to the site of the infection. Our criteria for CSF metagenomics is a little bit simpler. Basically, if all your preliminary testing, your tier 1 testing is negative, and you have high white count in your CSF and high glucose protein, then we'll send it. And so we actually felt like we were having an open door, but not a lot of people took us up on it just because the time was so long. Okay, all right, I've delayed all the good stuff, which is, of course, is looking at the data. So how have cases changed since we developed this policy? And so looking at the annual total of orders here, this is both CSF and plasma, and these are by orders placed and orders we actually sent. We actually implemented our policy just in the beginning of 2024. So pretty much everything in 2024 was regulated by this policy. And what we saw is that while orders continued to go up, ID was basically saying, we don't really think this adds value. And lab was saying, we agree with you. And usually the ordering provider would too. If there was ever a time where the ordering provider really felt like, ID and stewardship were not doing them a favor, I know we did have an escalation pathway, but really went that way. So we're feeling pretty good about it. In terms of plasma metagenomics, we also saw that the types of cases that we sent also changed. So kind of beforehand on top, before the policy, we're seeing a lot of febrile neutropenia. We're seeing a lot of vague pulmonary infection, and also just kind of a smattering of random things, pericarditis, necrotizing colitis, persistent fever. And we were sending more often than not. After we had this policy where we're making some recommendations on what we think is high yield and what we can get clinical action out of, we saw a big shift where we're seeing a lot more invasive fungal infection rule out. We didn't see much change in infective endocarditis. Interestingly, although we feel like the data is good there, our providers feel like they can manage it clinically using other criteria. And then a lot of those other things kind of dropped off. We sent less febrile neutropenia, but very few numbers. So this policy maybe helped people choose when to use it. We didn't say no that much more often, but I felt like we just got folks asking for different things now that they knew what they could use it for. We looked a little bit at, in total, how often we saw high yield results. This is actually looking at that by indication. So, invasive fungal infection is IFI, infective endocarditis is IE. We didn't really see any clear winners here. We saw a lot of blue being our insignificant pathogens. Interestingly, this pulmonary infection, we found a Candida albicans, and we said, well, we don't think that's causing a fungal infection. And then 2 days later, the patient was actually candidemic. And so this was probably a real result, but we actually didn't believe it at the time. So as a whole, we can still say our results are usually positive, but not that informative for plasma metagenomics. CSF metagenomics, here's the slide I told you we'd talk about. We sent 11 over the past couple of years, but we only had one positive, and it was a definite pathogen. It was an amoeba. But most of the time, we just got none detected. We weren't really sure how to get value out of that. We also saw that when we first started offering this a little bit more broadly, people took us up on it, and we were sending it. And now, our infectious disease specialists are saying, most of the time, it's not actually going to add value. I think a lot of these purples here, where we decided not to send, actually had to do with the realistic turnaround time of 2 weeks. And so we're excited to see what 2025 brings, where maybe we can actually have a result faster. One of the ones we sent in 2025, like I said, got delayed in a snowstorm, unfortunately. And then if you look at in total, over time, did our policy actually change? What was a good diagnostic yield or not? We went live in 2024, and we can at least say that we didn't interfere with the diagnostic yield we were already having, which may not be great, but it didn't go up, but didn't turn to zero at least. Okay, so outcomes after the policy, there's some things that can't really be quantified, which is, the things that can be is we sent fewer, actually, which was kind of surprising. The range of indications was narrower, which we felt good about because there were indications that we felt good about sending. We had much better retrospective analytic data. Now we can look back at all these cases. We have a lot more conversations to say, how did this help? How did this not? every time you send a test that doesn't help, everybody involved gets better about selecting the next one. We're really happy that our subject matter experts are the ones asking for the test and the ones interpreting the test. So ID is kind of taking center stage here. And then the improved communication really helped. In the very early days, nobody knew how long it would take to get a result. Nobody knew what exactly to collect. We'd get BALs collected for metagenomics. We'd get swabs from feet collected for metagenomics, all kinds of crazy stuff. But now it's very defined. We have a hospital policy you can look at and figure it out. So qualitatively, we felt much better about the process, and our teams, I think, were happier to be part of learning how to use this test. So as my last slide, just talking about some next directions for what we're thinking about. We'd like to clear more indications from any stewardship review at all. So right now, for us, the only one that's actually cleared is infective endocarditis. Invasive fungal infection feels like it's at that point where we have enough criteria, we've seen enough cases, we know that you can just go. It's not always going to give you a hit, but we know we feel good about that testing. And of course, looking for others as the data comes out. Can we clear them from stewardship review or add them as recommended indications? Part of it is providing more education to people who might order this test. So thank you for coming today. Maybe you learned something. Of course, with CSF, there's massive changes to the turnaround time, which is really going to change how we think of everything in terms of when we should send, and maybe we'll send a whole bunch more. So we're updating our policy right now. It's not quite out yet. There are still lots of low yield tests here. We're not getting a lot of new diagnostic value. And so we're still trying to figure out how can we figure out which patients will really benefit and ensure that we're not accidentally not sending the ones that would have actually helped. So this is our ongoing process, both reducing our low yield and increasing our high yield. And then we anticipate that there will probably be expanded specimen types available for testing commercially. I think BAL is probably the most logical next step. I'd love to see more sterile fluids, joint fluid, pleural fluid, things that you expect there not to be, or maybe you expect there to be many organisms, but you don't expect there to be normal organisms there. And so we'll see if those come down the line. Who knows? Maybe here at UW. I don't know if anybody in the audience has a spoiler. Anyways. And with that, I'll give you my acknowledgments and I'll thank you all. I really want to thank all the Plugs faculty, Anna, Lisa, and Jane for helping me review these in a timely manner. And a lot of times I had no idea a request was coming. And so getting those alerts so we could have that conversation, that huddle early, was really important. Sean Murphy was directing the lab with me for a good amount of time, and having to field these requests and talk to folks before we had a policy was a huge headache and really informed how this work should be. Our infectious disease colleagues generally have been really supportive of trying to do better with plasma metagenomics. And I really want to thank Dr. Stokes and Dr. Chung for helping come up with a lot of data for CSF metagenomics specifically. Lastly, the lab always gets the short end of the stick. I was constantly calling frantically to see if we could get a specimen out early, or to see if we had something frozen. And so the microbiology and reference labs teams at Children's did a wonderful job of keeping everything straight and being honest about what we can expect in terms of turnaround time. Okay, it went a little further than I thought, but we still have some time for questions. So I would really appreciate it, first of all, if you gave me all a review. With that, let's chat.
Can you comment on metagenomics versus broad range PCR functions?
Yeah, so that's a good question. We didn't get too much into the weeds on exact testing algorithms. Of course, with broad range PCR, you're kind of setting some parameters on what you're looking at. Broad range PCR is good when you're looking for one thing. But if you expect there to be a lot of things there, the identification of one organism might preclude the identification of a second organism. So this is helpful in those scenarios where you kind of expect there to be multiple organisms present. That's why nobody will ever do 16S PCR of the blood, or at least they should.
Can I ask about low yield versus no yield? I can't tell from your stories. Do you have examples where the testing really sent people down in the correct direction for either plasma or CSF, or are we at a zero right now? Just given the time of turnaround time and cost quantities.
Yeah, so for plasma, I think there are a couple wins that we have had. They're mostly actually drawn from negative results. So kind of pushing us the fact to discontinue workup or antimicrobials. It's really hard to do, particularly in very ill cancer patients where you feel like you have to keep on your empiric therapies because you don't really know what's causing their symptomology. there were a few that we actually got the answer, but we didn't believe it. And that might have been just because we're unfamiliar with the results. One notable case, we actually got a Corynebacterium diphtheriae out of a plasma metagenomics in a patient with a cardiac vegetation. And we're like, no way that's causing infective endocarditis. Eventually, we got invasive specimen collection, and it was indeed Corynebacterium diphtheriae, which we don't really think of as being an endocarditis pathogen, but it can happen. And so I think, knowing when to trust the results is a part of creating those no yield into high yield cases. And you need some basic literature to get there.
So on one of your slides, it mentioned that Delphi requires proprietary sample collection criteria and fluid. So as you're thinking about restructuring your workflow, how are you incorporating that? Because one of the things that seems nice is you can just sort of like, collect the sample anyway, we'll store it. But if there's proprietary aspects, that might become more challenging.
Yeah, so I'm sorry, I should have been repeating the questions this whole time for the online audience. The question is about Delv Bio's proprietary stability media. And I put that in there just because it's, a logistical thing that we have to know about, but they, will just give you a bunch of it and you can have it on hand. It's very shelf stable. So it's more of a question of, to your point, if you want to go for metagenomics, once it's in that buffer, you can't use it for maybe autoimmune testing. That's one of the values that we got out of, frozen specimens. You could say, hey, we'll freeze it. Maybe we'll use it for autoimmune testing. Maybe we'll find another specific PCR we want, or we could do metagenomics. Here, that kind of does lock you into that with that specimen. And so, yeah, it's a hard choice to make at the time, but usually I think a lot of the patients for CSF metagenomics tend to have enough where you could probably split the sample, but you got to know.
When you're talking about yield, I guess it seems like touched on a little bit in the question, but the focus on positive results versus negative results. And, technology is very specific, but the sensitivity question, I think, needs to be adjusted in trials. But also, is there like a biological limitation? Like this is oftentimes used as this, like, do we start steroids or do we continue empirics? And so even with additional studies, are we get to a point where a negative test is going to allow de-escalation of these indications?
Yeah, for the folks online, the question is, do we know how we can take a negative result and ever make it actionable? And I think the answer to your question is maybe not. This may be a case where metagenomics never fully replaces conventional testing. Instead, you have to do both if you want more information. Now, like we looked at, a lot of the time you'll get less overall yield if you've already done everything, but maybe the cost will come down. We're talking about thousands of dollars today, but who knows, maybe in 15 years we'll laugh and treat it like a PCR.
And then I have another question. Do you think there's a future for antimicrobial susceptibility information and from this data?
That is a great question. I'm optimistic. I think that what we know genomically about microbes, for gram-negative organisms, there are so many paths to developing resistance that we know if you find a carbapenemase that you're carbapenem resistant, but we know you can be carbapenem resistant without a carbapenemase. And so identifying all the areas that could lead you to resistance, plus really what we need is a little bit more information from metagenomics. How much coverage do we have of the organism? If you only are detecting like a little bit and it's not even like a quarter of the genome, I would not feel confident that we know anything about how it would react to antimicrobials. But if you have the whole genome 6 times over, we would probably know if there was a plasmid in there too. So I think that work has yet to fully be explored, but I think maybe one day we can get there.
I was curious about the logistics of this collect and hold policy when you gave data about increasing log MPNs in fungal infection, as well as the persistence of cell-free DNA, even in the face of antimicrobial therapy. So why is early collection actually part of the policy? And
yeah, that's a good question. So I think maybe I should have included more data there. The question is, why do we collect early? So I think fungi are the outlier, that when you, that potentially the amount of nucleic acid is going up over time. What we mostly see, and we mostly know about it from kind of bloodstream related infections, is that detection goes down. So for infective endocarditis, for example, there's a good cohort study where they show it's near 100% for the first few days, and then it persists longer than culture in terms of detected signal, but it starts to wane, and you can imagine that several months after your primary infection, maybe nothing. And so the other good question, and it does come up time to time, is, well, you got one in the freezer. You could collect one now, but you can never collect one 2 weeks ago. So it's better to have something and not need it.
Perhaps this question highlights a misconception about what I think the outputs of metagenomic sequencing is. But could you speak to the cases where you get multiple organisms from a MNGS test? And do you can you speak to can you use coverage as a weighting for prioritizing therapies for certain pathogens over others in a multiple basically multiple hits or multiple organisms from a sample, I guess?
So the question is, how can we use complicated polymicrobial metagenomics results to maybe come up with a better empiric therapy? And I think that is maybe beyond my personal expertise on how to do. Some folks are feeling that way, like we could, if you see a lot of stuff and you'd be like, well, we've got gram negatives, we got gram positives. I think what stops a lot of folks is that usually these patients are in critically ill states. And just because you don't see a gram-positive organism doesn't mean you can stop your gram-positive coverage, for example. And so I think it really is hard to, when you have something positive, to be like, can we stop or de-escalate? Now, maybe, if you're treating for amoeba and you find no amoeba in a CSF metagenomics, then you feel better about that just because, your testing is a little bit more narrow. But it's a difficult question. And I think if we had clinical outcome data, we might be able to answer that. If you just treated what you saw, do patients do better or worse? That's an open-ended question.
Thank you so much.
Thanks, everybody.